Hello, my name is Micheal A. Byfield from grade 1210. So we're planning on making the animal cell. Got illustration board, if you can see down here. Got some paint, more illustration board, bubble wrap, more paper. And got some colors just in case. Always need scotch tape. Just some other things, glue, scissors, or other art materials. So, bigger scissors in this, or glue, is all the stuff you need to make a giant recyclable cell. So we're planning on making the animal cell. All the materials, so let's get right into making this animal cell. So here's the first stage. I already drawn this out, so to make it a lot more easier, got the drawing from Google as usual. So I'm just gonna cut this out and make sure this is will be the foundation of this animal cell. Since I already cut this, the foundation of this right here, don't mind the cuts, but there's other materials that I have to use, so I got this box. Hey mate, you need it? So we're gonna make another template of this, so we can trace this and cut this out, so we can make this a little more creative and more 3D like. So let's get into cutting. You guys with things, especially can't even drive. Goodness. You already cut out the box that's been destroyed by me. So I'm gonna put the bubble wrap here by this foundation up here to make it a little more 3D like. So let's get to it. Okay, so I already cut this and glued all of this to put this on the foundation. Also, I drawn this as well, so I can start cutting this and put it on other cardboard so to make this more 3D like. So let's get right into it. I already glued most of this, but I'm gonna put this piece in right about, say right here. Other piece right here. Product of the animal cell that it's been 3D modified. So next we'll be getting into any one of these, the way how it works, what's the purpose, the
We have
functions.
4 of the basics of the animal cell. Got the nucleus right here, nucleus, the outer one. Then we got the ribosome, that's the outer blue. Then we got the vesicles, these green parts over here.
And after the 4 parts, they will get to individually getting to all the other parts and in the next detailed part, so be up to that. First up is the first one. We got the nucleus, which is the largest structure in the nucleus, consisting of nuclear organizers, chromosomes, RNA, and proteins. And the main function of this is primarily serves as the site of ribosome synthesis and assembly. And we got the nucleus as the other part of this is the notable within the cell, the genetic control center of the cell and chromium, and surrounds by nuclear envelopment. And the main function of the nucleus is to control protein synthesis and direct cell division and many metabolic activities of the entire cell. And we got the other part is the cell membrane, this the entire thing of the cell, which is the border of the cell. It's about 8 millimeters thick, and also a semi-permeable membrane which composed of proteins and lipids, and the unit membrane model arrangement of plasma membrane or protein lipid protein. The cell membrane is a fluid mosaic model. Protein molecules penetrated into the lipid layers and not continuous on the surface of the lipid. And the main function is to support and protect the cell, which regulates the movement of the material and in and out of the cell, and facilitate the diffusion scatter of particles from high concentration to low concentration. This is the endoplasmic reticulum. It has 2 types, which is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. This one is the hard. We'll start with the hard, and it's a large interconnecting membrane of the tunnels continuous with the nucleus envelope. And the main function is to make proteins or secretory proteins to make more membrane channeling products both in or outside the via membrane. And the smooth endoplasmic reticulum is a network of interconnected tubes as well. The main function of that is to synthesizing and scattering of certain steroid hormones, enzymes of carbohydrate metabolism, and enzymes of the lipid synthesis. And this is the Golgi body, named from Camillo Golgi, an Italian biologist and physician. Has a series of 3 to 20 parallel flattened sacs closely stacked together called cisternae. And end of the sacs are bud off curious vesicles, and the main function of these is receives and modifies and packages the substance manufactured by ER. This right down here is, of course, the mitochondria, a spherical rod-shaped structure from 0.2 to 7 millimeters, a double-layer membrane, cristae or complex folding of inner membrane, which is made of, as mobile structures and capable of changing their shapes. And also, of course, 5th graders knowledge is the powerhouse of the cell. And the main function is just to produce energy of form of ATP. And the next part of the cell is the lysosomes, which is produced by the rough ER and the Golgi Proteus, which 2 Greek words is breakdown body, and are membrane-bound, dense appearing structures that contains enzymes or acid hydrolysis. And the main function of these are that act as waste disposal units in digesting and removing foreign materials, or also known as a suicidal bag. We got the vacuoles, which is an enclosed compartments which are filled with water containing inorganic or organic molecules inside enzymes in solution. And the main function of this one is to digesting, also a storage of chemicals, cell enlargement, and water balance. Okay, next is the ribosomes, which is tiny spherical structures, and the bodies are in which the amino acids are bound together and a site for protein synthesis. We got the centrosomes, which is quite near the nucleus bud, and in the side of the centriole is a pair of small rod-like structure. They are attached by microtubules in the wall,
And the main function of this part is to activate and process all of cell divisions or mitosis.
Well, folks, that's the end of this video. We have discussed and make the animal cell and discuss about the parts. So this will be the closing in. So thank you for watching and see ya.